Alright, hi folks. We are now in Lab 7. Welcome back, and we're going to talk about factorial treatment structures. I'm not going to really go over the first couple pages of the lab, but you should read it. There's a section here stressing again that factorials are not experimental designs, but they just refer to how levels of treatments and factors relate to one another, and so we can have factorials within any of the experimental designs we've talked about so far. After that, on page 1 is a little bit about coding for interactions within R, within the LM function, and so using colons to partition out specific interactions, asterisks to do all-inclusive combinations of factors, and then using the caret to specify an upper bound to how complicated an interaction you would like to include in the model. So take a look at that. On the next page, I talk a little bit more about how factors factorial treatment structure underlying a treatment, how they relate to one another, and that the degrees of freedom all add up. I talked about that in class as well, but here's another presentation of that. What I want to do is get right into example 1, and here we are dealing with an RCBD. It's a two-way factorial, so we have two factors. One factor is varieties of mustard, and so we have genotypes, or varieties, VAR, I have 5 of those. And we have 3 different soil mixtures, soil, and we have 6 pots for each. So we're dealing with a situation where we have 6 replications, it is blocked, and we have 5 levels of one factor, 3 levels of another, so we have 15 total treatment levels. And because we have 6 reps of those 15, that gives us the 90 experimental units in our study. In this particular experiment, we are interested only in those 5 varieties and only in those 3 soil mixtures. Our conclusions will not extend beyond those to things not included in the study. So we regard both variety and soil as fixed factors. So let's go ahead and I brought that into our studio here. So here you have Lab 7 Exercise 1. I refer to the data set as must.dat for mustard varieties. And when you bring the data set in, you see there's 3 classification variables, soil and variety, the combination of which creates one overall treatment classification variable, but we partitioned it out into the underlying factorial structure, and then the block that it's in. So we have 6 blocks, we have the 3 types of soil and the 5 varieties, and then here are the yields. So once you bring in the data set, you want to ask about the structure of it, and in this particular case, soil is running from 1 to 3, variety from 1 to 5, and blocks from 1 to 6. So R thinks they're all integers. So lines 10 through 12 here is we are reassigning them as having factor status, and we look at the data set again, and now we have our factors where yield is a number, so that all looks good. The ANOVA is pretty straightforward. It is a old school RCBD linear model in which the response variable is explained by block and treatment. But now in this case, treatment, I'll type it right here, our treatment is soil, it is also variety, it is also soil times variety. And that's what this soil star variety is shorthand for. It's soil plus variety plus soil times variety. So I'll get rid of that. So that's the model. We run the ANOVA, and here's the table that we get. We see, in fact, that our soil star variety actually was shorthand for 3 different lines of the main effect of soil, the main effect of variety, and their two-way interaction. With this initial ANOVA, because it's a factorial treatment structure, your first stop in all of this is looking at the interaction. And here we see, even though we're asking a very general question, we've taken the sums of squares of the interaction, dividing by 8, and asking if that average portion is significant. It is significant. We are below our threshold, our type 1 error threshold, so we conclude there is an interaction. And that's when we're sort of forced along a simple effects post hoc analysis rather than a main effects analysis. Now, to do that, first thing we want to do, because it is an RCBD, we want to make sure that we're meeting all the assumptions of the model. You could grab your residuals from here, and you could run a Shapiro test. You could run a Levene's test for each of the 2 factors. You'd have a Levene's test for soil and a Levene's test for variety. All that syntax looks the same as what we've done before. Here on lines 24 and 25 of the script, what I'm doing is running an exploratory model to look at the two-way block interactions with each of the varieties. And you'll see here I have block plus soil plus variety. I have the three classification variables in my model, but now I've enclosed them in parentheses and I followed it with a caret and a 2. When I do that, what you'll see is that in addition to soil variety and soil times variety that were in my original model, it's included two other terms, block times soil and block times variety, both of which are non-significant. So it's telling me that my soil and my block are acting additively, which is what I would like to see, and my variety and my block are adding additively. This is the kind of thing that if we didn't have the degrees of freedom, we would have to be probing using a Tukey 1 degree of freedom test. But because we do have the degrees of freedom, so we still have 40 degrees of freedom left in our error, we are able to pull these terms out and take a look at them directly. Now what would happen if we didn't cap this? Let me remove this. Let's look at all possible interactions between soil, the block, and variety. So I'm not going to cap it anymore at 2. So I've just put a bunch of stars in between. If I do that and run the model, R gets really mad at me because I've exhausted all my residuals. These 40 degrees of freedom for the three-way interaction are the 40 degrees of freedom here in my residual term. So this is a case where we are actually using the three-way interaction as the error term in our model. And so we're assuming that three-way interaction is non-significant. And if you were really interested, you could try to visualize that three-way interaction the way we did it in lecture. But you can take some solace in the fact that because the two-way interactions are non-significant, it is very, very, very unlikely, and it happens very, very infrequently, that you would have non-significant two-way interactions and then suddenly a highly significant three-way interaction. So these non-significant two-way interactions are giving you some confidence that these residuals, the three-way interaction, is actually a fairly decent approximation of what the true experimental error is in this particular study. Okay, so now we know, I'll return this the way it was. Block plus soil plus variety, and I'll raise it back. I'll cap it at 2. Okay, so we know from the original ANOVA table here that we have a highly significant interaction between our two factors, so therefore we have no, we are not justified in characterizing the effect of soil on yield ignoring which variety we're talking about. In other words, we really have to go variety by variety and characterize the effect of soil. And then conversely, we have to go soil type by soil type in order to talk about differences among varieties because of this interaction. The way we do this is the way I was showing you in lecture day. We want to subset the data. So lines 29 through 31 are how we subset the data according to soil type. So here I'm creating a smaller data set I'm calling must soil 1, in which I'm taking all the experimental units that received soil 1. And the way I do that is I subset the original data set, selecting only those rows in which the soil variable equals 1. I can do it for soil 2 and soil 3, and then with these three different subsets of data, I can run ANOVAs on them. So I can run an ANOVA on all the experimental units in soil 1, and the way I do that is I have a very reduced model now. Soil no longer appears in my model. I'm only dealing with 30 experimental units now. Right? Originally I had 90 experimental units, but only 30 of them received soil 1. But because all of those 30 experimental units are homogeneous in terms of soil, soil is no longer a distinguishing factor in my study. Now this is just a simple RCBD in which block and variety appear. And that's true for soil 2 and for soil 3. So these three subsets of data are now fed into 3 linear models, 3 different ANOVAs. And then what I could do is, if I were interested in ranking the soils from best to worst, I could then run individual Tukey tests on each of those subsets of data. And so here you see the Tukey test that I'm running on each of those subsets of data. So let's look at all of that. So if I'm going to create all my subsets of data, and now so I called it must soil 1. That's one of them. Let's head that and just take a look at that must soil 1.dat. You can see that the only guys in this subset of data are those that receive soil 1. So soil 1, as I was saying before, becomes a non-distinguishing factor in terms of the yields that we have here. So it essentially drops out of the equation, and my linear model only includes the 2 informative factors that remain. So I have my 3 data sets. I can run my 3 ANOVAs. Bump, bump, bump. And here you see them here. This is the ANOVA for soil 3. There's the ANOVA for soil 2. There's the ANOVA for soil 1. And what's interesting is that it's only, so in soil 1 I see no differences among varieties, in soil 2 I see no differences among varieties, but in soil 3 I see significant differences among varieties, and that is the interaction. If I wanted to run Tukey mean separations, I need to activate my Agricola library, and now I can run my 3 different Tukey tests if I want to see any one of those. So let's look at Tukey, whoops, don't want to type it there, I want to type it down here. Let's look at Tukey soil 1, and there we have it. And what we're seeing is that there are indeed no differences among varieties. That's what we saw on the original F test. I could look at Tukey soil 2, I see no differences again, but when I look at Tukey soil 3, suddenly differences appear. So these are the simple effects of variety at each level of soil. And then, of course, you can take it from the other direction. I could subset the data by variety. So now I have 5 varieties, so that means I have 18 experimental units in each of these subsets of data. I can create those 5 subsets of data, 1 for each variety, and then I can run my ANOVAs for each of those subsets. Now, just like soil dropped out as an informative factor when we subsetted by soil, variety now drops out as an informative factor when we subset by variety. So now we have an RCBD in which only block and soil appear as factors. And let's go ahead and do all that so we can create all the subsets. We'll run all the ANOVAs, and let's go back and look at them. So here, for variety 1, we see a marginal effect of soil. For variety 2, also kind of marginal. Variety 3, it's a little more significant. Variety 4, it gets more significant by an order of magnitude. And variety 5 is also slightly significant. So the significance bounces around just a little bit. So it seems that all the varieties, that soil seems to have an effect no matter what variety, but that it's not all the soils. It's only really in soil 3 that we see differences among varieties. And you, of course, can run Tukey mean separations within each level of variety, like we did before, and you can summarize all that output. So I think that's probably what I did. Okay, so here on page 3, I talk a lot about the degrees of freedom of the interaction, what's being used for error and what isn't. You should take a look at that. And yeah, so here on page 4, I've gone through and summarized the results of all of that, looking, subsetting by variety. This is the effect of treatment, the effect of block, what the mean significant difference, minimum significant difference is, and what the Tukey tests would show in each of those cases. So I'm going to stop right here because I got to keep it under 15 minutes to get it up on YouTube, but I will start the next one with visualizing the interactions in this particular example.